Hello everyone. Welcome to the biotechnology series in which we are doing advanced tools and techniques, and I, Dr. Charu Dogra Rawat, Associate Professor from Ramjas College University, will be talking about DNA fingerprinting today. So the learning objectives of the session would be to understand the principle and procedure of DNA fingerprinting and to know and talk about various applications of the DNA fingerprinting technique. Now, when we say DNA fingerprinting, it is known by several terms: DNA profiling, DNA typing, genotyping, genetic fingerprinting, genetic typing, identity testing. All these terms are basically synonymous and they refer to the technique of DNA fingerprinting that we would be discussing today. It is a technique that simultaneously detects many markers in a DNA or a genome to produce a pattern unique to that DNA. Now, what does that basically mean? It simultaneously detects many markers in a DNA genome. So, we are analyzing the DNA for certain markers which we are detecting simultaneously, and that the detection of these markers produce a unique pattern for that DNA. We will be elaborating on it more further. Procedurally, the DNA fingerprinting includes two biotechnological techniques, and these two techniques we have already covered, which I briefly tell again here so that you can understand the DNA fingerprinting better. And these two techniques are, one, the restriction digestion, and the second is southern blotting. So procedurally, the DNA fingerprinting includes basically these two techniques. Now, what do we understand by restriction digestion? Now, please recall that restriction endonucleases, the type 2 restriction endonucleases, they cut the DNA molecules at specific nucleotide sequences in the precise, reproducible manner and result in blunt or flush ends or sticky and cohesive ends. So, they are basically nucleases, that is, they cut the DNA molecules. Endo, because they cut in between the DNA molecules, and they cut at a very specific site. So, they are termed as the restriction sites. There can be two parts of the restriction sites. One is the recognition sequence and another is a cleavage sequence. So, depending upon what type of restriction endonucleases is used, it recognizes a particular site and it either cleaves inside that recognition site or adjacent to that recognition site, etc. But that is basically what restriction endonucleases are. Now, what do we mean by that they produce blunt ends or they produce sticky or cohesive ends? Then again, please recall the restriction enzymes, for example, ALU1, when it cuts the DNA, then on both the strands, it cuts at the same position. So, what you get is, as shown in the figure, these kind of blunt ends. So, these are blunt in origin after the cleavage by the enzyme such as the ALU1. So, they produce the blunt ends at the end. However, the sticky ends are basically when an enzyme such as EcoR1, HIND3, BAMH1, they cut the two strands of DNA at different positions. So, when they cut at for example, at one of the strands it is cutting after G, and on the under strands it is again cutting after G, but because it is complementary, then it is basically asymmetric cleavage that is happening on the two strands. And therefore, you can see that these two kind of ends where you have one overhangs, these are called as overhangs. So, you have got these overhangs that are produced. They are termed as sticky or cohesive ends because when you will bring these two molecules together, then because of the complementarity of these overhangs, they tend to stick to each other. They will not become double stranded because there will be no synthesis of the phosphodiester bond here, but still they will come and they will be kind of, they behave as like magnetic. So, they are sticky to each other because of the complementarity of these single strand overhangs that are produced. So, these are the two kinds of ends, and we understood in detail how this restriction digestion is a basically step in the molecular cloning or the production of a recombinant DNA. Then how it is ligated and then how it is transformed into a host where it makes multiple copies, and that is termed as the molecular cloning itself. But basically the idea is that to talk about, because this DNA fingerprinting involves this restriction digestion. So I just reviewed that how the restriction endonucleases cuts the DNA molecule and produce either the blunt ends or the sticky or the cohesive ends. Now when you run it on a gel, how does they appear? So for example, this is just an example gel photograph. So you know that we can visualize the DNA fragment by separating them on the agarose gel electrophoresis, where you apply the electric current, and based upon the where the positive side is, the DNA is going to run towards that because DNA is negative side. So this is basically a gel which is representing the restriction digestion of a DNA which is in the lane 2. Now lane 2 is an uncut DNA, but when you cut the DNA, then you are obtaining these two fragments. So this is probably a little bit of undigested DNA that is there, but these two fragments are created. And when you run it with a DNA ladder or a marker, so what is a ladder or a marker? Again recall, these are basically the known sized fragments. So any DNA, for example lambda DNA we generally used, so lambda DNA when they are cut with restriction enzymes, so they are also cut with restriction enzymes only. So when they are cut with restriction enzymes, we know that what base pair size do they produce, because we know the genome, and then we locate the sites of particular restriction enzymes, and then we can say these are the fragments that we got. So these are the fragment sizes. So when we run our digested DNA with a known sized DNA fragments, then we can actually correlate, because we started simultaneously, we can correlate that this particular size, what will be the size by comparing it to the ladder DNA. So this is how basically it is done. So you can see that the addition of these two fragments, the base pair, is going to give rise to the size of the uncut DNA which is basically there, or a linearized DNA which is there. So this is how you analyze. The idea to tell here is that basically the restriction enzyme can cut the DNA into fragments, and these fragments can then be separated on the agarose gel electrophoresis. So that is what the idea which is required to understand the DNA fingerprinting. One point another which is important to note is that the restriction sites are generally not evenly spaced out along a DNA molecule. So when we see a DNA molecule, for example just shown here, you can see there is a couple of enzymes, there is a BGL1, BGL2 and other enzymes which are shown here. They digest the DNA, however, their restriction sites are not equally spaced. So they are not equally spaced. That means that if you cut with one of the enzymes, they are going to produce multiple sized fragments. So one thing important is to note that the restriction enzymes digest and cleaves the DNA molecule at specific sites. Second important thing to note is that they produce the fragments and it can be visualized on the agarose gel electrophoresis. Third important point to remember is that these are not equally spaced. So when you cut with a single restriction enzyme, then it will cut all the sites and will produce multiple sized fragments. So again, as shown here, for all the three enzymes, based upon the sites, they can produce multiple size fragments. Now why this multiple size fragments they are appearing? Because what happens is, when you are doing a digestion, then it is not necessary that the digestion is complete. Usually when there is a partial digestion, then what you mean by partial digestion is that the restriction enzyme is going to recognize one site and cleave it, but going to miss the another one. So that another one, it is going to cleave in another molecule, because DNA, there are number of molecules that are present. So in some of the molecules it will cut at the first site. For some of the molecules it will miss that first site and will cut at the second site. For some of the molecules it will miss these two sites but cut at the third site, and this is the largest site which will remain uncut. So when you do a partial DNA, then you basically get all the different sized fragments that you have got. Or even if, like, for your digesting it with the same enzyme, so even if it is a complete digestion, here it is showing that it is a complete digestion. So what happens in complete digestion? You get different sized fragments, because this is the largest one. For example, we are considering the blue. So this is the largest one, then this is the smallest one, then smaller, then this is the smaller, this is again the smallest one. So you will get different 4 or 5 kind of fragments which are basically there. So the total size is this, and the different sized fragments are this that you are going to get. So in partial digestion, it happens when it misses it, but in complete digestion this might be the different sized fragments because they are not equally spaced. So these fragments can then be separated and visualized on the agarose gel electrophoresis. So these are the three important points basically that needs to be remembered. The other technique that is involved in the DNA fingerprinting is the Southern blotting. So we know now what was the Southern blotting. Again, this picture just recalls what we have talked in detail about the Southern blotting. What happens in it is that when we run the restriction digestion gel, so they are going to give different fragments, we basically blot it onto the nitrocellulose membrane, then we fix it on the nitrocellulose membrane, we make it single strand, and so this is basically how it is transferring to the nitrocellulose membrane. So we create these kind of stacks, or we do electrode transfer, or whatever the way, but basically all these fragments. Now in this manner or in electrode transfer manner, the exact pattern that you have got will be kind of a photocopy on the nylon membrane which is here. So the same fragments are going to, in here it moves by the capillary action. So on the nylon membrane what you are getting is the exact pattern which was on the gel. So the same pattern you get, then you make it single stranded, you do a number of washes, and then what you add is the probe. So probe was basically an identifying oligonucleotide which is going to go and bind to the gene of interest, and therefore it is going to give rise to some kind of a signal. If you are using a radioactive probe, it is going to give rise to the radioactive emission, whatever that wavelength, it is going to be developed on the X-ray film, or if it is a colorimetric, then it is going to be giving a rise to a colored reaction, and again producing a color that can be counted on the colorimeter or spectrophotometer, or basically can be seen as the bands again on the X-ray. So it depends upon the detection mechanism that is used. But the idea is that in all these fragments, if you want to identify a particular gene of interest, or if you want to identify a particular pattern, you add the probe. Now depending upon the probe you are getting, you will get the entire pattern basically copied onto, or whatever you are going to detect onto the whatever the detection system is. For example, X-ray here. So the probe has identified only these things. So when you run the marker, you can add the marker probe also. So you will get a series of marker lines, and then you are going to get the pattern for the genomic DNA. So this is another technique which is involved in DNA fingerprinting, and that basically is a two-step procedure where first we do the restriction digestion, we load it onto the gel, and then we identify, or we basically, in that we are not, in DNA fingerprinting usually we are not identifying a single gene, but we are basically identifying the entire pattern of the genomic DNA. So we can use a particular probe, and therefore it is going to give rise to a particular pattern in different DNA which is basically there. So what happened is, so these are the two techniques. I just want to review these two techniques really quickly, because then you understand procedurally how the DNA fingerprinting is done. Now when I said what DNA fingerprinting is, I used two terms which are very, very important. One is a marker. So we said that simultaneously we detect the markers that give rise to a unique pattern. So these two are the important terms to understand the principle behind it, or how are we going to basically have this kind, why it is called as the identity testing, or why it is called as the genotype profiling or genotypic thing. Why? Because we are going to create a profile, we are going to create basically a pattern of DNA based upon whatever probe we are using. And the important thing is that that pattern would be distinct, that pattern will be unique, and that is why it is termed as a fingerprint, because you know that in fingerprints, our fingerprints are unique except for the twins. The fingerprints are very unique. In fact, it is used for identity testing. If you want to identify an individual, you record their fingerprints and you have a database, then you can actually match those fingerprints and it can very precisely tell you what the individual is. So similarly, here rather than the fingerprints, you are creating a pattern of the DNA, and in fact the DNA also, this pattern is going to differ. Why? We will see just in a short while. So no two individuals produced by sexually reproducing organisms except for the identical twins have exactly the same DNA. Although we have the same genetic content, which is the DNA, our hereditary material is the same, But for every individual, even between the siblings, even between the parents and the, children, even between the relatives, for every individual, this DNA is not the same. And why? We know that it is basically we are all Homo sapiens, so we can carry the human genome. But again, there are variations which are there. And what are the reasons for that? There is a reason. Why? Because there is a recombination that occurs. So we have done this in molecular biology and genetics a lot, that there is crossing over of chromosomes in meiosis prophase 1. So because of this crossing over of chromosomes, our DNA recombines, and therefore the children does not have exactly the same DNA as their parents, because while during the meiosis prophase 1, the recombination occurs, the crossing over occurs. And you understand why this is so important. Why? Because evolutionary, this has got a very, very much evolutionary significance, because if by chance something wrong happens in a particular gene and there is no variation in that gene across a population, then that entire population will be eradicated. So because of the adaptability, because of the evolutionary aspects, this recombination is a very, very important thing to happen. So this is why the DNA is distinct between individuals. Then there is random alignment of maternal and paternal chromosomes in meiosis metaphase 1. So you know that one set of chromosomes we get from the maternal side and one get from the paternal, from the father, and when these mother and father chromosomes, they align themselves, then there is a random alignment between them. So after random alignment, there will be crossing over as well, but whenever there is a random alignment, then that means basically this randomness is going to create the variation. Then of course there are mutations that occur. So the mutations, there can be spontaneous mutations, there can be mutations because of the agents which are there. So all these mutations, which are basically insertions, deletions of regions of nucleotides, they all are going to cause the differences in the DNA between the individual. Similarly, there can be DNA replication errors. So when we did DNA replication, and we also did the DNA repair mechanisms in molecular biology sessions. So this DNA replication is not a foolproof method. There always is a chance and there occurs the errors in DNA replication. Many of the DNA replication errors are repaired if they are particularly very consequential, then they are basically repaired by the repair machinery, but still some go away, some do away with that, and therefore they also have the same effect as the mutation, that is the variation or the difference in the DNA would occur. So these are some of the reasons that cause, or that is the responsible for the variations in the genome that we see. So these genome variations can be in the form of single nucleotide changes, it can be in the form of insertions or deletions, it can be in the form of all different kind of things that can arise in the DNA. So based upon these variations, what we do is we identify certain variations that we can use as markers to generate a unique pattern. So you see how the DNA fingerprinting story is developing. So first we understand that the DNA of individuals are not the same. Then we understand how these variations occur, and now these variations we can actually use them as markers. Because they are different in different DNA and genomes, if we use them as marker, then we can actually identify the unique pattern, which is basically for a particular individual, and we can use it to distinguish individual from individual. That is the entire principle of DNA fingerprinting. So what are these types of markers that we use? So what are these changes? What are these variations that we use as the markers? So they are, for example, RFLPs. We will talk in detail about what these RFLPs are, that is these restriction sites. So we talked about the restriction sites, that these restriction sites are not equally placed. So when they are not equally placed and when they are distinct in different DNA, then because of the variations, these restriction sites can be generated or they can be removed. So because the two DNA are not identical and because of the reasons we said, the recombination, the mutation, the replication errors, these sites can be formed or these sites can be dissolved. They cannot no longer act as the restriction site. So because of that, what we are going to get is the restriction fragment length polymorphisms, which is RFLPs, and these RFLPs can be used as then markers. So we can use these RFLPs, we can analyze these RFLPs in different individuals, and because they will produce unique pattern, we can use it for DNA fingerprinting. Similarly, the length polymorphism detected by PCR. So this is detected by restriction digestion, but we can also detect, so this is the another technique that can be, or that is basically involved in DNA fingerprinting as well. So there is a restriction digestion and southern blotting, there can be PCR also, and that also can give rise to a different pattern. We do not need the detection of southern blotting in that, we will talk about this in detail, but basically the length polymorphism detected by the PCR also can be used. There are allele specific oligonucleotide probes. So if you remember, we talked about these allele specific oligonucleotide probes in our last session, where these probes, and then we used primer specific for the amplification as well, and we said that these are basically specific for the gene amplification. They are not random primers we are using. If you remember, and you can go back and check the quantitative PCR sessions, there we talked about the allele specific oligonucleotide probes. So in fact, they can also act as markers, because one allele will be distinct to another allele, and because of the distinction we can use the specific probes that are basically there. And then there are the markers, which are the repeated DNA. So under the repeated DNA, there can be mini satellites. What are these mini satellites? Mini satellites which are termed as variable number of tandem repeats. Now you know that human DNA, there are a lot of repeated DNA. In fact, when Human Genome Project was there, and when finally the entire human genome was sequenced, the scientists were astonished to find that there are only around 30,000 genes which are there. And there is a lot of junk DNA, and that junk DNA constitutes is basically the repeated DNA. So there are repeated elements of DNA that are basically present. So if you have got a small block, for example, repeated units of several 10 base pairs basically, so what happened, these 10 base pairs are going to repeat one after the another. So 1, 2, 3, 4, 5, basically blocks of repeated units can be there, and these blocks can vary between the DNA of the two genomes. For example, I may have a particular unit, for example AGCTGC, and this ACGTGC is repeated 5 times in my DNA at a particular location. However, in my sibling's DNA, it can be repeated 6 times or 7 times. So when we are going to create a digestion pattern, then we are going to see, and if for example a restriction site falls beyond this unit of 5 units of or 6 units, so in my case the size of the fragment will be 5 unit long, but in my sibling's case that will be 6 unit long. So that tells us that that is a distinct DNA from what I have got. So these are the mini satellites or variable number of tandem. Tandem means one after the another. So variable number of tandem repeats. It was discovered by A.J. Jeffries in 1985, who basically gave the DNA fingerprinting technique also. So there can be microsatellites. So in microsatellites, they are short tandem repeats. There are repeated unit of 2 to 6 base pairs. So they are 10 or more base pairs long, the mini satellites or microsatellites, which are very small, or which are termed as the 2 to 6 base pairs long, which are there. So these are the repeated DNA. Now there are certain marker selection criteria which is there. The one thing is that the markers should be polymorphic, so that they are informative, so that they are going to produce many forms basically when we use these markers. The markers should be single locus, that is, they are only present at one position, because if they are present at multiple positions, then we are not able to distinctively see what the pattern is. So that they occur in only one location and there is no ambiguity about their number or the position. Other criteria involve that the markers should be neutral, so that they are not correlated with selection or adaptation unless selection or adaptation are to be studied. So we do not want that there is certain kind of a bias in their selection or adaptation which is there. So the neutral markers are basically used, otherwise they will be basically favored to be selected. The markers should be located on different chromosomes so that the markers are independent. So they are not, genetically transformed in a linked manner. So we understand about the linkage. So because in linkage it is not going to produce a true result, they have to be present on the different chromosomes so that they can be independently assorted. So these are basically the simple criteria for choosing a marker, and we talk in details about the marker and the applications of DNA fingerprinting in our next session. Thank you.